So if they will not join, Pooja, we can start for it. I mean, these are the recorded class so that the student can actually see after this class. So let's give them 1 or 2 minutes. So meanwhile, what we can do is instead of just sitting, we can revise few things so that if they will join, we'll get to know. Can you see this slide?
Yes,
Pooja,
sir.
can you? Okay. So yesterday we saw about splitting the cells, right? We talked about how when cell will reach to a certain confluency. I explained the concept of confluency. Then what we need to do is we need to split the cells. Uh, and we generally split when cell will reach to 80 to 90% confluency. For splitting, we would require some reagents, and one of the major reagent is the trypsin EDTA. This particular reagent, or it is again helps in detaching the cells from the layer monolayers from the petri dish. It is pancreatic protease and basically cleaves this lysine and arginine residue. Then we talk about the specific protocol that we use for splitting the cells. So what we do is first of all we check the cells confluency if they are in proper confluency or not. After that, what we do is we remove all the media because the media contain FBS and that can inhibit the action of the trypsin. So we should remove all the media, wash the cells with PBS so that all the residual media will get removed. And after that, we need to add trypsin, 2 to 3 ml. And then we put this plate back into incubator at 37 degrees Celsius. In 2 to 3 minutes, what will happen? Due to trypsin, all cells will get detached, and we can collect these cells into Eppendorf tube, allow it to centrifuge, so that all cells will get pelleted, and they will be come at the bottom in the form of pellet. Then we can add the fresh medium, and we can split the cells. I talked about the splitting ratio of the cells. And you like now, right? If you are splitting the cells into from one plate, we are making four plate out of it. Then that ratio is known as 1:4, and based on that, we can prepare the plate that we require. Uh, I believe we uh, we started with this thing, how we can freeze the cells, and for that. I talked about the media that we generally use. It is known as freezing media. The specialty of this freezing media is that it contains DMSO. And the function of DMSO, we saw why it is necessary to add DMSO. It basically helps in preventing the formation of water crystal inside the cells by increasing intracellular solute concentration. And due to that, the ice crystal will not form inside the cells and cells will be prevented. So this is the composition of the freezing media that we generally use in lab. Some lab have luxury to have this commercial freezing media, but most of the lab use this composition. So the composition, as we talk about, we use 5% DMSO and about 40% serum in incomplete media, and that media can be used for freezing the cells. Then I talked about the effect, like we cannot suddenly keep the cells at -20 and -80. During that process, what may happen, or we suddenly cannot keep cells into liquid nitrogen. So that will also affect because there will be cold shock if we will allow cell to go to that lower temperature. So we need to gradually decrease the temperature, and for that purpose we use this box known as Mr. Frosty. cryo. It basically have this isopropanol in this box, so that the temperature decrease will be 1 degree Celsius per minute. So gradually its temperature will decrease. Now what we'll see the protocol for uh, freezing the cells. The reason of this freezing was, like, as we were taking this example, your experiment got over. Now you want to store the cells for longer duration, maybe for 2 hours, 2 years, 3 years, or more than that. If you want to store the cells for longer time, or in general also, if you want to store the cells for longer duration, we use this freezing of the cells protocol. Okay, so first of all, what we need to do is, we should uh, we should clean the lamina hood. That is the very first thing. And again, as we saw for all other experiment, we should keep the medium, the complete media, trypsin and PBS at room temperature at 37 degrees Celsius, so that their temperature will be at 37 degrees Celsius. Okay. And in addition to that, freezing media will be there. So we can take out that freezing media. Generally, we store at minus 20 degrees Celsius or sometime at 4 degrees Celsius. So take out that medium, keep it at 4 degrees Celsius. That is, then it is okay to keep it at 4 degrees Celsius. Now we'll start our splitting the cells protocol. So first of all, if, you will observe the cells under microscope. If they have reached to certain confluency up to 80% or 100%, and what you will do, you will remove all the media that is present in the cells, and wash these cells with PBS, just like for passaging cells you were doing. Apply, you should wash the cells and add trypsin on the monolayer of the cells, and keep this petri dish or plate inside the 37 degrees Celsius CO2 incubator. So at this temperature, what will happen, your all cells will get detached, and next step is the stopping the trypsinization procedure. In last lecture, I told you how we can stop the trypsinization procedure by adding complete media, right? So 5 to 6 ml of complete DMEM we will add, and mechanistically how it will happen, how that reaction will stop. Basically, your complete medium contain FBS, so that FBS will stop the action of the trypsin. So just add 5 to 6 ml of trypsin, stop this trypsinization reaction, and collect the cells into a sterile tube. Okay, and centrifuge it for, let's say, 3000 RPM for 3 minutes. So during this centrifugation, what will happen? Your all the cells will be collected at the bottom. Then you can remove supernatant, and in this pellet, now add the this freezing media that we prepared, which contain 5% DMS and 40% FBS, and mix it nicely. And at at the same time, what you need to do, you need to uh, keep this cryo vials ready, label them properly, because um, these cryo vials maybe will be used for after 2 years or 3 years. So you won't be able to see check which kind of cells are there. So that's why label these cryo vials properly, like by saying the name. For example, the HeLa cell is there, then write down HeLa. Below that, write down the passage number. Like while passaging, every time the passage number get increased. Like for example, today you passage the cells, so it will be first passage. After 2 days or 3 days you are passaging, that will be passage number 2, and so on. So you should write the passage number, and below that you should write the date on which cells were freezed. After adding this 1 ml medium or 1 ml suspension of the cells into this cryovas, close the lid and keep these vials, cryo vials, into Mr. Frosty. So that frosty, if whenever you are keeping it into Mr. Frosty, what will happen? The temperature of this surrounding will be gradually decreased by 1 degree Celsius. Keep that Mr. Frosty box into minus 80 degrees Celsius for 24 hours. Okay, during this temperature time, what will happen? Temperature will gradually decrease to 1 degree Celsius, and all these vials will have temperature minus 80 degrees Celsius. After 24 hours, on next day, transfer that box into liquid nitrogen, and the after transferring it into liquid nitrogen, you can store your cells for longer time. So this is the protocol for freezing the cells. So I will show you one video of how exactly we perform this freezing the cells. So it will get clear to you. Just hold on 1 second. Yep. Yeah. I will share my screen. Okay. So these are the cryo wells. Can you see? Pooja, can you see?
Yes, sir.
Okay, perfect. So in this cell culture we saw, right? I mean, all the protocols. So first of all, what she did, she cleaned this surface with 70% ethanol. This cryo wells she opened and kept it for UV light, and after that, these surface was also given UV light treatment so that there won't be any contamination. She took out these reagents and kept at 37 degrees Celsius, which is a complete medium trypsin and PBS. So that is essential for trypsinization of the cells. So take out these all cryovials, label them. It is very essential to label them based on the cell type, then the passage number. This P is basically what she's writing is passage number and date. So this is Mr. Frosty that I was talking about. This is simple small box, a plastic box inside which the metallic C. Inside this metallic ring is present. So now let's say these are the HEK293FT cells are there. And below that you can see P14. P14 is nothing but it is passage 14 number. And below that date is there. So now if you want to store this cell, how you can do it? Just take out these cells and remove the medium, liquid medium from it, the complete media. And discard it. And now wash the cells with PBS. So give a gentle washing. And discard the PBS as well. Now add trypsin. So she's adding 2 to 3 ml of trypsin into this medium. Into this plate. And spread it and incubate it at 37 degrees Celsius. Stop the trypsinization procedure by adding this complete DMEM. So during this process, you will actually able to see the cells are detaching from the surface. I mean, first of all, it will be kind of turbid or that kind of surface will be there. But when this trypsinization process will get over, you will able to see that the cells are actually getting detached from the surface and they are coming into your solution. So this is how you should flush that particular part of the petri dish or this particular cells with the help by taking this medium and flushing on the cells. And now collect the suspension which contain your cells into fresh tube. So you can either perform the cell counting as well, that is optional, and centrifuge it at, let's say, 3000 rpm for 3 minutes. So this is the cell counting procedure. This, yeah, whenever you count the cells, there will be this chamber, the cell counting chamber will be there. So what you do is this blue color dye, this is known as trypan blue dye. A special type of dye is there, which will not be uptaken by live cells, which will be selectively taken by uptaken by dead cells. So from these, you combine dye. See, you can see these are the this is cell counting in the chamber. So what you are observing here, these are the different boxes here, and this white round shaped thing is the your cells, and there are some dead cell as well. So you can count by just calculating all the cells that are present, and there is this one formula to calculate total number of cells. So this is the white palette. Can you see, Pooja?
Yes, sir.
So this is your cells, and this is the media that you want to discard. So discard all these media, and now resuspend these cells into freezing medium. So can you see? I mean, obviously this is from thermo. So they have this commercial option, like, I mean, the commercial media, commercial freezing media is present, but in lab also you can prepare the freezing medium. So just resuspend cells into freezing medium. Can you see how gently she is doing? So that and now add 1 ml of resuspension into this cryo vials and keep your cryo vials into this Mr. Frosty. So this Mr. Frosty, if you see inside this isopropanol is there. I mean, just this upper metallic part can be removed, and you can pour isopropanol inside it. So just keep all your cell like this and close this Mr. Frosty and put this into minus 80 degrees Celsius. So this is this refrigerator is basically for minus 80 degrees Celsius. The other box is also there, but she kept it. So you will keep it for overnight, right, at minus 80 degrees Celsius. And on next day, what you will do, uh, you will take out this Mr. Frosty and transfer it into liquid nitrogen. So this is the liquid nitrogen. Take out the box and transfer your vials into this liquid nitrogen. So these are the cryo gloves that specifically, you must use while handling the cells. So just take out the cells and transfer into a separate box. And put it back into liquid nitrogen. So this was the procedure for freezing the cells. So I hope you understood this, Pooja. Do you have any doubts about this protocol? Like, I mean, how one should do, or in any step you have any doubt?
No, sir, I understood, sir.
Okay, perfect, perfect, Pooja. So what we'll do is we'll see next one very important technique that is known as transfection. At least we will start with that technique, uh, and then Maybe tomorrow or tomorrow we can again continue it.
Sure, sir.
Okay. Okay. Just now we discuss about various cell culture protocols about reviving the cells, passaging the cells, and freezing the cells. So all the experiment that you perform in molecular biology or on cell culture. So Pooja, why do you think I mean, I have explained this, but why do you think that it is necessary to perform this experiment on cell culture, mammalian cell culture?
So the cells are used for different purposes like drug testing, sir.
Yes, yes, right, right. And also we can
Other
perform, yeah, go ahead.
other cloning experiments, sir.
Yeah, that cloning part also, that experiment also can be done. But the first thing you told me is like, I mean, we can treat the cells with either drugs, or what we can do, let's say a very interesting condition. You want to see, okay, the a gene is there, okay, any gene is there, very novel gene is there, and we will consider this hypothetical condition in which your boss or your guide PI ask you, okay, Pooja, what you do is you check what is the function of this gene. A
Okay, sir.
novel gene is there. You want to identify the function of that gene. So how you can perform? There are like two approaches by which we can study the function of the genes. Either what we can do is we can knock down that particular gene, knock down or knock out. That is loss of function. Just remove that gene and see the effect of knockdown in the cells and compare that with control one. The second approach, what we can do is we will overexpress that gene. Okay, and we'll see if its expression is getting high, then how it is affecting. So these are two approaches. When we are overexpressing that particular gene, so it is known as gain of function, and we are removing it or deleting it, doing knockdown, that is known as loss of function. Okay, so in that way we can perform this experiment and we can identify the effect of that particular gene. Okay, now let's say if you want to study the very important function of gene, which is your favorite gene? Give any name of any gene. Can you think about any gene?
Cryo, sir. Cryo.
Which one?
Cryo, sir. CRY.
Okay, CRY gene. Okay,
Yes.
okay. So you want to identify its function. So what you can do is you can selectively target that gene by there are different approaches, knockdown approaches, and you'll delete that gene or do knockdown. And there will be these two condition, Puja. One will be the control one, and the other condition in which you removed or deleted that gene, and you will observe the phenotypic difference, and also you will see how what are other genes got affected due to knockdown of this gene by performing qPCR or western blotting. And there are different assays are there, like DNA damage assays are there, then proliferation assays there, and a lot of assays are there. Then you will see, okay, when I am deleting this particular cryogene, so the expression of, let's say, p53 is getting upregulated. So, and apoptosis is happening and DNA damage is happening. So you'll say, okay, I depleted this gene or I deleted this gene, and due to these pathways were affected, so suggesting this particular gene may have a role in those pathways. Okay, so for those purposes we basically perform knockdown experiment to identify its function. Is it clear, Pooja, why we basically perform this experiment and how we can check its effect or its function?
Yes, sir.
ठीक
Yes.
है, okay, okay. So there are these two approaches. by which loss of function can be studied. First one is the knockdown, and the other one is the knockout. Okay, so very basic difference between these two techniques or method is knockdown is basically what we do is we reduce the expression of that particular gene by certain percentage, let's say by 80%. So what it means that we give some siRNA or LNA gapmers so that its expression, if it was 100-fold in control condition or in normal condition, then upon knockdown its expression will be only 20%. Arbitrary, if it's in normal scenario, if 100 copies of these genes are present, then we will target 80 copy and only 20 copies will be present there. So this approach is known as knockdown, whereas knockout is this approach is completely removing that gene. So there are these CRISPR-Cas9, shRNA. These are approaches are there. So with this approach, what we can do is we can completely remove that particular gene, and there won't be any expression of that particular gene. So how basically this knockdown work? Uh, so if you, let's say you want to perform knockdown of this mRNA, cry mRNA, and you want to deplete its expression. So what you will do, you will design siRNA or LNA gapmers against specifically to your target gene, which is CRY. Okay, and its sequence will be exactly complementary to this mRNA. This will be around 20 base pair. So this 20 base pairs will be complementary to your gene. They will bind here, and degradation of this mRNA will occur. This siRNA will again it will work in different fashion, and LNA gapmers will they have their different mode of action. So will talk about their mode of actions. First of all, let's see how this RNA silencing and siRNA basically works. So this siRNA, how they get processed, this double stranded RNA with the help of dicer, it will get converted into siRNA, and AGO and RISC complex will get loaded, and these siRNA will specifically bind to mRNA target. This one is the mRNA target, and this AGO and RISC complex, what they will do, they will allow degradation of this mRNA. So let's say this was your this was your CRY mRNA, which having this some sequence. So we will design siRNA against this CRY mRNA, and we will use, so with the help of this AGO and this siRNA specifically will bind to CRY mRNA. Complement, these bases will be complementary to each other, so they will specifically bind to this mRNA, and due to presence of this RISC complex, degradation of this mRNA will occur, and you will see knockdown effect. This was about siRNA. Now we'll talk about LNA gapmers. How basically uh, we can use this LNA gapmers to perform knockdown study. So LNA gapmers, I mean, it could be a very long lecture about LNA gapmers, but if I want to put it into very simple and very short form, so this LNA is basically locked nucleic acid, and these are the modified oligonucleotides. The chemistry between these base pairs is modified one as compared to the siRNA, and their efficiency to perform knockdown is superior to siRNA. Okay, because they are modified one, so that other enzymes will not degrade these specifically, and they will be protected one. So how this LNA gapmers basically works? So can you see this two color here? This the pink kind of, and this yellow color is there. So this pink region is basically, these are the DNA, single stranded DNA, which is locked in between these two sequence. Okay, so that's why its name is locked nucleic acid. So this DNA, single stranded DNA, is basically locked in between these sequences. and the sequence of these LNA gapmer will be complementary to your target mRNA. So let's say transcription is happening of cryogene, mRNA is forming, like all the processes happen, splicing and everything happen, and your target mRNA got formed, crymRNA. So what you will do, you will use LNA gapmers. So these LNA gapmers are complementary to your target mRNA. So they will go and bind to this target RNA. Now there is this one enzyme is known as RNase H. Okay, so this RNase H, and you may be write it down. The what is the specific or what is the specialty of this RNase H enzyme is that whenever there is hybrid between DNA and RNA, these RNase H will degrade it. So you can see this blue color, it is your target mRNA, and this pink one is the DNA. So here this hybrid is formed. So this hybrid will get recognized by RNase H, and that hybrid will get cleaved due to this presence of this RNase H. And in that way, your degradation of target mRNA will occur and knockdown will happen. So with the help of LNA gapmer, you can achieve up to 80 to 90% of knockdown. And the first one that we saw, siRNA, with this you can able to get 50 to 60% of knockdown. So that's why these LNA gapmers are superior. Okay, but this is not that much straightforward, like you're using LNA gapmers and completion knockdown happen. You need to use some reagents uh, in order to process, carry this experiment. So there are two reagents that will be required to perform this experiment. The first one is the lipofectamine. This is again carriers for your LNA gapmers or siRNA. These are the lipid vesicles basically, and their structure you can see it here. Like different types of lipid bilayer molecules are there. Like this dotma is there, dospa is there, and transpectam. So that this long chain obviously suggesting these are the lipids. And the reason why we need to use this lipid is, so you can see, okay, for example, this is the nucleic acid, and this is LNA gapmers, what is shown here B. So you want to use this siRNA or LNA gapmers against a mRNA, and you want to target it. But when it will get into cells, so this RNA will get this RNA or DNA will get cleaved by cells machinery. There are RNA-CH and DNAs are there. So what will happen? These RNA will get cleaved there, and it won't be able to target your mRNA. This siRNA and LNA gapmer will get cleaved here. Additionally, it is not possible for nucleotides to enter the cells. So these are two inhibitions are there, two hindrances are there. Um that's why we cannot directly use siRNA or LNA gapmers. We need to have some kind of vesicle or transporter molecules. So what these transporter molecules will do, they will form these carrier molecules will form these vesicles, small vesicle will form. And inside these small droplets of lipid, LNA gapmer or siRNA will be present there. And these are the basically positively charged lipids vesicles. So after that, your LNA gapmers or siRNA will be packed inside it, and now these combination you can use for transfection. So they will protect your it is present inside of this vesicle. So it will basically protect your siRNA or LNA gapmers from all the enzymes, and additionally it will allow to pass these complex inside the cells. So that's why use of this lipopectamine, which is RNA-MX, it is essential. When it will go to cells, then this LNA gapmers can be or siRNA will come outside, and it will target the RNA, and knockdown will happen. So this is how this lipofectamine will look like. This is one example of lipofectamine 2000 uh, RNA IMAX that we basically use for knockdown experiment. The other reagent that you would require for your knockdown experiment is optiamium. So this is like, I mean, as the name suggests, optium. Again, this is medium only, just like DMEM or MEMM. But in this media, what is specialty of this media is that in this media the serum level is reduced. The FBS that we are going to use, its percentage is reduced, and by what percentage they reduce, we do not know, because it is their proprietary, so they will not disclose it. So this is the commercial one, optium medium. So these two reagents we required in order to do transfection procedure: optium and lipofectamine, these two reagents. So this one is the reduced one. Now we'll go to protocols, how we basically perform this knockdown of any mRNA or any genes. So Pooja, is it, I mean, you're understanding it,
Yes, sir. Understand.
or should I go slow, or make very basic you
No,
know
sir. It's fine.
okay, perfect. So this is the protocol for knockdown experiment. Uh, what you need to do is, whenever, like, I mean, let's say you want to perform knockdown experiment for cryogene. You want to deplete its expression. So what you will do, you will first of all start your experiment in cell culture. You will take two petri dish in which you will add the cells, and after, or like, I mean, on next day, you will observe the confluency of the cells. So when cell will reach to confluency, let's say 40 to 50% or 50 to 60% confluency, it means the surface of the petri dish is cover up to 50 to 60%. That is the good starting point, because if cells are more, then there is possibility that a transfection will not happen properly. If the confluency is less than that, less than 50 to 60%, then also what will happen, it will have negative effect and cells may die. So generally, what we try to do is, we try to keep the confluency at 50 to 60%. Okay, now you're performing this experiment. You took two dishes. One will be the control one, and the other one will be knocked on. You should label it. So to start the experiment, as I said that first you should, when like, I mean, whatever the experiment, whenever you're performing or working in any experiment, first of all you should read the protocol and you should keep ready all your reagents. So for this particular experiment, and you can write it down, what is necessary that your optiamium should be at room temperature. So generally we keep this optiamium at 4 degrees Celsius. So what you will do is, you will take a small alicode, small volume of optiamium, let's say 4 ml or 3 ml, into a fresh falcon tube, seal it, and put it at 37 degrees Celsius, so that it will be at room temperature and cells will not experience any cold shock. So that first thing you should do. And it goes without saying that you will clean the laminar hood, and switch on the UV light, and take out some appendrop tubes. These are the appendrop tube of 1.7 ml capacity. So take out some appendrop tube and open it and give UV light to this also. So this is these are the things that you should do prior your experiment prior to your experiment. So that you will do. Now we'll go towards the actual experiment, that how you should do it. So just take this two tubes. One will be the control one, and the second one will be knockdown tube. In both the tubes, what you will do, like I mean, one tube will be for control siRNA and other will be for your specific target siRNA or LNA gapmers. uh, for example, cryogene. So label them and add 250 mule of optimum in both the tubes. In this tube also you will add 250 mule, and in this tube also you will add 250 mule. Okay. And now in control tube, add these uh, you know like I mean, there should be these four tubes that you should prepare for one single control and one knockdown. There you will require two tubes. Pooja, am I making sense here? So for
Yes, sir, but I didn't get why the four trips.
yeah, so that's what I wanted to say. So for like I mean, it will come into this like I mean, in subsequent protocol it will come. So for control, let's say you should take two tubes, and for knockdown you should take two tubes. Okay. So first tube, in this first tube, what will happen? I mean, just take consider these two tubes. Okay, this is for, let's say, your gene. Okay, and similar two tubes are there for your control condition. So in all the tubes, you will be adding optimum. That you should add, and in one tube you will be adding lipofectamine that we discuss. This one, lipofectamine, you should add, and in another tube, in that 250 ml, you will be adding LNA gapmers. So this gapmer will be either control one, or it can be specific to your gene. So now it is clear, Pooja, why we are using four tubes.
Yes, sir.
So for knockdown also, we will use these two tubes. In one tube, you will be using lipofectamine, and in another tube, you will be adding LNA gapmers against specific genes. So for that reason, there will be four tubes. Two tubes will be for control, and two tubes will be for your gene. So in this condition, 250 ml optimum you are adding, and in one tube you are adding specifically lipofectamine, which is a carrier for your siRNA, and in another tube you are adding either control siRNA or siRNA against specific genes. And now in like after adding lipofectamine or the siRNA, close the lid, tap the tube at the bottom, so that complete mixing of lipofectamine or LNA gapmer will happen. Just close the lid and hold it, and just at the bottom with your finger you can tap it 3 to 4 times, and allow it to settle for 5 minutes. Keep it at room temperature for 5 minutes, and after 5 minutes, what you need to do is, there will be this two tubes, right? In which 250 ml optimum plus siRNA will be there, and in another tube, 250 ml optimum plus lipofectamine will be there. After 5 minutes, what you should do is open this both the tubes and add this 250 ml of reagents in these tubes, drop wise. So during this procedure, what will happen when you are adding lipofectamine plus optimum into another tubes? Small vesicle, round vesicle of lipofectamine will form, and those vesicles basically are needed to carry your genetic or this siRNA. So you can see this is the representation of this lipid vesicles. So a round shape, this drop had formed, and inside that your siRNA is present. So this is very much important, that you will take all the reaction from here, or the 250 ml optimum plus lipofectamine, and add in second tube drop wise, and again close the lid of the tube and mix again, tap it. You may centrifuge it for like I mean, giving short spin, and incubate it for 20 minutes. So during this 20 minutes, what will happen? This complex will form of your lipid vesicles. Inside that, your siRNA will be present there. So during this 20 minutes, it will happen. And after 20 minutes, what you can do is uh, discard the media from your petri dish in which you want to perform experiment, and in that plate add 5 ml of optiamium. So the reason of doing this is that when you will add this optiamium, so cells will go under some kind of stress, and they will uptake whatever present to their surrounding. So because your optiamium contain less serum, so that's why these cells try to uptake whatever present in their surrounding. So you will add 5 ml in this plate, and after this 5 ml of optiamium, now add the reagent that you mixed. That you prepared, 250 ml plus siRNA and 250 ml plus lipopectamine. You have mixed into a single tube, right? And you incubate it for 20 minutes. So add the solution from this tube into petri dish drop wise. So this basically each drop will contain these lipid vesicles. Inside that, your siRNA will be present. So these small droplets will be spread out throughout this plate. And now this siRNA will come outside, and they will target this will target your gene. So you should keep it for 24 hours inside the incubator, so that what will happen, your siRNA will come outside and it will target the genes and knockdown will happen there. So this is just again illustration how basically it is done. Let's say you want to perform this experiment in the cells. Take these two tubes which are shown in green and blue color. In one tube, what you will do is you will add 250 ml of optiamium, which is a reduced medium, and in that you will be adding lipopectamine. Take another tube, these tubes are for one condition, like control condition. Similarly, you will do for knockdown condition, same procedure. Take another tube in which add 250 ml of optiamium and add siRNA. Allow it, tap it and allow it to settle for 5 minutes. After 5 minutes, what you can do is transfer this lipopectamine into siRNA dropwise and allow it to incubate for 20 minutes. So during this 20 minutes, your lipopectamine and siRNA will come together and they will form this complex. And after that, you can add this two mixed solution in your cells dropwise, so that the complete addition of your siRNA will happen there and incubate it for 24 hours. So after 24 hours, complete knockdown will happen. Pooja, got it clear? Is it clear to you? Hello? Can you hear me, Pooja? Anyways, time is over now. So what we can do is we will meet tomorrow, and in tomorrow also I will try to explain it again, and we'll see how we can perform gain of function or expression. So chalo, bye bye. See you tomorrow.